gracias a todos por acercarse al seminario de Dept. Pues fue compañero mío en la Universidad de Cornell, donde hicimos el doctorado en Ecología y Evolución. Dan, después de la Universidad de Cornell, hizo un postdoc en Michigan y actualmente es profesor asociado, hizo el postdoc en la Universidad de Berkeley, y actualmente es profesor asociado en Michigan, en la universidad, y también es curador asociado de las colecciones de herpetología y reptiles, anfibios y reptiles. Dan está interesado en preguntas relacionadas con biodiversidad, preguntas tales como por qué hay algunos clados que tienen muchas especies, otros clados que tienen menos cantidad de especies, cómo se forman las especies. Y habiendo venido a un departamento de ecología y evolución, sus trabajos también tienen un componente genético y un componente ecológico, lo hace súper interesante. Dan además es una persona muy viajada, un explorador nato. Durante los estudios en Cornell, si recuerdo bien, muestreo en Australia en una de las peores olas de calor del año, donde los canguros, como me contabas, caían muertos directamente. Ahí trabajó varios años. ¿Continúas trabajando en Australia?
Ah, sí. Sí.
Y actualmente también trabaja en la Amazonía peruana. Así que, y es un fanático también de observación de fauna, entre ellas las aves, con lo cual se salva. Bueno, los dejo con él para que les cuente mejor su...
Gracias. So I'm actually, and we're living now in Puerto Madryn on sabbatical. We'll be there until August. So Conocet pride here. Well, thanks for having me. So it's fun to be here. This is my first time in Mendoza. So I thought I'd talk today about just kind of general things that we've been thinking about in my lab and they kind of bear very generally on this idea of how predictable macroevolution is and how microevolution connects to macroevolution, which is something that I came to, thinking about out-of-date websites. But I think back to like the very first website I made in Irby's lab when I was a PhD student. When I first met you in like 2003, I was like, I'm really interested in microevolution connecting to macroevolution. So in some ways, that's where we still are. And before I start, I was going to ask people to think about in their mind, if I say, think about adaptive radiation and think of a couple groups that are undergoing adaptive radiation, or what you think of from your learning so far, what groups pop into your mind? So I think for lots of people, when you ask them this kind of question, the things that pop into their mind are the things that you read about in textbooks, like, anola lizards from the Caribbean, which undergo these, diversification in different islands into these ecomorphs associated with different kinds of ecologies and different parts of trees or bushes. Or you might think of things like Hawaiian honey. Often we're thinking about, island groups that have undergone these evolutionary explosions of morphology and ecology and so on. But, one of the starting points that I think is worth keeping in mind is that even though we think of these textbook examples as illustrative of adaptive radiation, I think it's really good to remember that these patterns are everywhere, and they are not just in island systems. You don't have to look very far to see examples of cases where species diversity and morphological diversity are really different, or dramatic, and also where those patterns are really different between groups. These are representatives from different radiations of Australian squamate reptiles. So you have things like elapid snakes and agamid lizards and one granid lizards and so on. And all these groups have to some degree undergone some degree of evolutionary diversification or radiation within Australia. But within this group, there's actually within these groups, there's a tremendous amount of variation in how successful those radiations were in terms of how many species they produced. So if I was to take all of those radiations and rank them by their species diversity that they have generated in Australia, you've got groups like Australian sphenomorphine skinks with almost 300 species. It's really diverse. And then you've got this long tail where you've got a bunch of other groups that have barely diversified at all, and some that have, even colonized Australia without even undergoing a single speciation event. So that's happened many times. So why do you have patterns like this? And at the same time, there's also a tremendous variation, amount of variability between groups and how much morphological variation they contain. So these are New World coral snakes right here. So I don't know if there's any coral snakes in Mendoza, but I would imagine they're close if they're not actually here. So
Yeah.
So coral snakes, if you've seen a coral snake, you know they're strikingly different in terms of their color pattern, and they're very beautiful. But a lot of that color pattern is, you know, it's a signal associated with advertising the fact that these are dangerously venomous snakes. On the other hand, if you take away that color pattern and you look at how variable they are, coral snakes are for the most part quite similar. And so there are species that are more ecologically specialized within the group or dietarily specialized, but to a first approximation, they kind of all have the same general body shape, and they generally eat the same things too. This is actually one of the rare species that does something a little bit different. This is an aquatic coral snake, and it specializes on more aquatic things. So, but you could contrast these patterns. This is a group of elapid snakes. This is a group of, you know, within coral snakes, I don't know what, there might be 70 or 80 species of New World coral snakes, and to a first approximation, they're all quite similar. You could take a look at other elapid snakes elsewhere in the world, like elapid snakes from Australia, closer, also elapid snakes. And this group has very different patterns morphologically. So this group has a lot of classically famous dangerous snakes like taipans and brown snakes, which you're seeing here. And these things, you know, kind of look like, are kind of morphologically similar, but there's a tremendous number of other forms within this group that are very different. So you have things like this death adder, which is an elapid snake that has evolved a specialized morphology within this group that looks very similar to a viper. It's convergently evolved with vipers, or both so jararacas, you know, here that you would find here in Argentina, for example. There are also a bunch of closely related to these big dangerous things like taipans you have, or brown snakes. You have these tiny harmless burrowing things that are within that same group. So the shovel-snouted snake right here eats different stuff. This one eats eggs of other reptiles. You have this tiny little burrowing snake right here. It's a specialized predator of other tiny legless lizards and so on. So all this variation has arisen in a very recent radiation of elapid snakes. So on one hand, you have coral snakes without showing much variability, but they have a lot of species. Australian elapids, closely related, but tremendous amount of morphological variation. So why do we have these patterns where some of these groups have so much variation in their species number? The very classic problem in evolutionary biology. And one of the ways we as a field have approached thinking about this pattern in terms of cause is to think about the idea that there must be, well, there is a cause, there is a pattern, there is a difference between these clades and what they have done. Well, a lot of our paradigm, the paradigm we use for thinking about that is that there is some kind of key trait or a key innovation that must underlie this differential evolutionary result you get. Maybe it's something like the classic example here is the idea of the evolution of a pharyngeal jaw system in cichlid fishes. Cichlid fishes have undergone these dramatic radiations in African lakes. And this pharyngeal jaw system is something that pops up in a number of fish groups, but it's especially elaborated in cichlids. So they use the oral jaws here. Normally fishes use their oral jaws for prey capture. Well, cichlids have evolved this special secondary jaws deep in the throat from their high branchial apparatus. And the secondary jaws, the pharyngeal jaws, take the role of prey processing. And so it frees up their oral jaws to essentially be to allow them to specialize for capturing diverse prey types, or for scraping algae off of rocks, or for crushing mollusks, for capturing other fishes, and so on. So it's sort of a key innovation that's allowing them to enter new adaptive zones. And the pattern in cichlids is, just to give you an example of why people, one of the patterns that this is proposed to explain is that cichlids really have done something really dramatic compared to other groups of fishes that live in the same lake. So these are cichlids, these are families of fishes living within Lake Malawi, and the number of species they contain. And despite all of these things living in the same lake and having the same geographic and ecological opportunity to diversify, only cichlids have done anything there. So really there's a real pattern here. So you can't just say, cichlids just live in a special set of environmental circumstances that promotes diversification. because these other fishes all had the same opportunity to do so, and they didn't. So that's why people end up looking for things like key innovations to explain those patterns, so organismal traits. What we're going to do today is we're going to look at some of the natural history of macroevolution in squamate reptiles and ask whether we can use key innovation type traits, or any type traits, to predict diversification. And then we're going to talk about mechanisms for variation in the rate of speciation in groups, also focusing on whether there might be key traits that affect why some groups are speciating more quickly than others. And we'll see if we can tie that back to some general lessons about predictability and macroevolution more generally. The only real background you need here, other than having a love for squamate reptiles and lizards and snakes, because you will see a lot of pictures of lizards and snakes throughout this talk. I hope you are not horrified by snake images, for example. Some people are, apparently. So the main background is that I'm going to talk a lot about rates of speciation. Don't need to know a lot about rates of speciation. We're not going to talk about the methods for doing anything with rates of speciation. But one of the things that we do in my lab is we build statistical tools for taking things like time-calibrated phylogenies. This is a phylogeny for New World warblers. And we take things like time-calibrated phylogenies and you run them through these machines of statistical or mathematical machines that we develop, and we turn them into evolutionary rates. So you might imagine turning this phylogeny here into a perspective on rates of speciation. This is really all you need to know, is that in this case, the colors are proportional to an estimated rate at which that branch and along that phylogeny is splitting at any point in time. And think about the endpoints here, these tips of the tree, as essentially being a rate of speciation associated with individual species. So we were to look at like what those are implying. So these are sort of the rates of speciation at the tips of the tree. You know, some species belong to clades that have relatively fast rates of speciation, and some clades belong to groups that have relatively slow rates. So obviously these are not independent of each other. If you're going to do any statistics on these things, you need to think about the fact that they're all sort of co-estimated and things aren't independent of the clade to which they belong. And then we're going to talk a little bit about how traits predict things like rates of speciation. And the only thing you need to know here is that any results I'll show you in this respect are using specialized statistical tools that account for some of the weird and non-independence or the correlations between data points that come up when doing these kinds of analyses. There's a lot of special tricks that we essentially have to do to adequately to analyze these data in a statistically fair fashion. But well, again, I won't talk about details. I'll just say my lab does work on methods for dealing with some of these sort of statistical weirdnesses that come up. So let's think about these squamate reptile story here. So what we're going to do is we are going to, and this is a large project that involves especially a couple of former folks from my lab, Pascal Teitel, who's now at Stony Brook University. He was a PhD student of mine, and Sonal Sengel, who was a former postdoc of mine. So we're going to build a giant phylogeny for squamate reptiles using genomic data. We're going to generate a bunch of genomic data and do it. We're going to date the phylogeny using the fossil record. Then we're going to use that to estimate evolutionary rates of things, like rates of speciation. We're going to put together a bunch of trait data that has involving everything we can get that we think might be affecting the of squamate reptiles, by which I mean lizards and snakes throughout this talk. We're going to put together morphological data on skull shape, for example. We're going to put together ecological data on diet, and we're going to estimate the rates at which different clades are evolving those types of traits. And we're also going to look at how different clades are different or differ in their magnitude of innovation. And when I say innovation in this sense, think about like I'll use this word over and over. When I think about innovation, the way I like to think about the ancestral squamate reptiles, some 250 or so million years ago. When I think about innovation, I'm sort of thinking about how much change has happened between that point and and the present day. So if I say, this clade has more innovation than another clade, I really mean that this clade, you say, has changed a lot more relative to what that ancestor is. And a non-innovated one by that metric would be a species where the morphology is quite similar to what that ancestor is. So, we could argue about that specific definition, but it is kind of objective and we can actually do some things with it. And so we're going to ask whether, how much variation is there in these macroevolutionary rates within squamates, and to what extent is it predictable or repeatable with respect to the evolution of particular traits. So then we'll talk about the details here associated with phylogeny, reconstruction, and so on. But we did generate a lot of data. We generated genome-wide data for about 1,000 species. Then we took GenBank data for another 5,500 species, and we put this all together in a big file. Did a bunch of used a bunch of fossil calibrations to date the tree. And this is sort of a snapshot view of the tree. You'll see this tree over and over again. Now the full tree has about 6,400 species, I think, in it. And here I'm showing you essentially a snapshot view of the tree. So I've collapsed this tree down to a representative set of 246 clades. So there's actually a number of species within each of these clades. But because it's just unwieldy to look at a tree with thousands of tips, this is an easier thing to look at. And so you got snakes over here, and these are lizards over here. And actually, even though snakes are lizards, so snakes are nested well within lizards, and we can all see this, I will nonetheless refer to snakes and lizards as separate entities in this talk. So if I say that, when I say lizards, I am referring to this paraphyletic thing over here that is lizards. And when I say snakes, I'm referring to this group over here. So what do some of these traits look like? So this is looking at this range of morphological and ecological traits and other traits that we have. So for example, an example of a trait. This is one of our supplementary tables showing some of these traits. These are the number of vertebrae. So we can look at like X-rays of a snake like this, and we can count the number of pre-sacral vertebrae. Or we could classify species based on their foraging mode. So some species are active foragers and they move around very actively and look for prey items, and others are what we call sit and wait predators, where they essentially sit, rely on camouflage, and come to them. It's a classic sort of dichotomy in reptile foraging behavior. We could also look at, this is a trait called the elongation index, and this is essentially the extent to which an animal is a long, skinny tube, or whether it's a short, fat thing. Now you might say, well, that's really obvious. It's just going to have snakes, all long, lizards, all short, fat. But in fact, within lizards, and things get really interesting here, within lizards, there are a whole bunch of groups of lizards that are not snakes that have evolved a snake-like body form. These are a couple of representative examples. So snake-like body forms, sometimes with limbs, sometimes without, have evolved multiple times within these other lynx, within geckos, within annielid lizards, within anguid lizards, and so on. So this has happened over and over again, this evolution of a snake-like body form. And we put a lot of effort into this particular set of traits right here, which is multivariate diet. In some ways, this is my favorite, this whole thing, and thing I'm most interested in right now. And so for about 30 separate dietary categories, we tried to collect as much data as we could, both from museum specimens on our own, as well as from published data sets that other folks had, and by bringing a number of collaborator data sets, where we tried to collect as much possible data as we could from primary specimens. So what we're doing here is we're not saying, the literature says that this species is a carnivore, this species is an herbivore. We're using individual level data where we can match an individual specimen to what was in its stomach. So that data set involved about 69,000 individual animals, over 1,300 stomach content data for that animal. So we know exactly what they were eating. It also lets us say things like, we know how much variation there is in the diet, for example, across species as well. So this is really the first result slide from this work. And what I'm going to show you here is just what is when you get to the level of looking at the trophic structure or the dietary structure of space across squamate reptiles. What do we see? And this is just looking at this 69,000 individual, 1,300 species, 30 prey category dietary data set. And to reduce that down, what we've done is we've done some principal components analysis here on the prey categories to reduce that down into just 2 PC axes. And we're, on this first axis here, each data point is going to be a species. And on this side, and this side of the scale is going to reflect whether things eat more invertebrates or more vertebrates in their diet. This up and down direction is going to tell us whether their prey tends to be more terrestrial or more aquatic. And this is just the first two PC axes. Each data point is going to be a species. The size of the points will be proportional to the number of individuals that we, actually the size of the points is going to be proportional to the width of the individual diets, the dietary breadth. And so blue is lizards and red is snakes. So you can see right away a couple of things. The big things that you see is that despite the fact that snakes are lizards, right? But they're nested well within lizards. What snakes do, they form this giant cloud that takes up a huge amount of the dietary space, whereas lizards essentially are constricted to, for the most part, to one region of that dietary space. So there's a really big expansion of dietary space associated with snakes. The other thing is that lizards also seem to be quite restricted in that space. And so snake predators are doing things like eating a lot more vertebrates, but they have a broad range. There's still some snakes that eat invertebrates. Snakes sort of span this entire spectrum of very aquatic prey to very terrestrial prey and so on. So, and we might ask, what's going on in this area of trophic space over here where snakes and lizards overlap? So, are the snakes and lizards, when snakes and lizards overlap, are they eating the same things? So, what I'm going to show you here is a way of breaking down these data points in here. And so it turns out that the species of snakes that overlap with lizards in that area of diet space are eating things that the lizards generally aren't. So this plot is actually masking a lot of complexity. So when you really look at the dietary specialization of the snakes and lizards in that plot, what we see is something that looked like this. The species of snakes, this is showing looking at those individual data points and telling you whether the snakes that are there are, how they are divided relative to lizards. So on average, the snakes, for example, have fewer adult insects in their diet. So this is saying that adult insect category is enriched in lizards. For spiders, it's enriched in snakes. Centipedes enriched in snakes, and insect larvae, pupae, and eggs is much enriched in snakes. So basically, there's a bunch of the snakes that seem to eat invertebrates, or arthropods in particular, focus on eating larvae and pupae and eggs. Whereas if you're a lizard, you're generally eating adult insects. So, and the other thing is these dangerous, large, hard-bodied arthropods that tend to be big, scorpions, large spiders, centipedes, those tend to be things that snakes don't. And there's another category of invertebrates that I'm not mentioning here that snakes often feed on as well, that you can see on this previous plot, where some of these, I think they're covered up in that particular view, but another dietary category that snakes tend to eat that lizards don't, that's an invertebrate, are gooey things, so snails and slugs in particular. So what I'm going to show you here is the difference in dietary breadth between snakes and lizards. And so this is one of the things we had to worry about with any kind of comparison. So there's a classic idea that snakes are more specialized than lizards in terms of their diet. But one of the big problems with that comparison is that people have always looked at more data for lizards. And so if you look at more data for lizards, you're going to be encountering more kinds of prey items. And we see that signal in our data. Average number of stomachs that we looked at per species for lizards is 45, and for snakes it's only 7. Snakes are a lot rarer in museum collections than lizards in general. And so we developed some special statistical methods that let us deal with this interspecific variability to get a statistically unbiased perspective on specialization. And when we do that, we see that snakes are, in fact, much more specialized than lizards. So it is true that on a per species basis, the average species of snake is much more specialized than the average species of lizard. So here I'm going to show you rates of phenotypic evolution and rates of niche evolution. So this is how fast species are evolving new dietary states and how much innovation we think they've had. So what I'm going to show you I'm going to show you data points for individual clade reflecting the average rate for all the species in each clade. And I'll show you rates for two things. One is this net innovation, change from the estimated ancestor of squamates to the present day. And the other is just going to be how fast they're evolving new dietary states. When we look at this, The only thing you really take home is that red points that are shifted to this side mean fast, and blue points that are shifted to this side mean slower or less. Lizards tend to be slow and blue, and this is for innovation, so less innovation. Snakes, fast, high, and red, more innovation. So their diets are more different, have shifted more from what we think the ancestral squamate was. Same is true for evolutionary rates. So snakes, faster and red, lizards, slow and blue. So relatively slow rates of dietary evolution for lizards relative to snakes. So, yes, slow rates in lizards, fast rates in snakes. And this general pattern holds true for essentially all the traits that we looked at in this paper. So that includes, for example, the rate of body elongation evolution. Even though there are legless lizards in their bodies, on the whole, lizards aren't doing this anywhere near as quickly as snakes. This is skull shape evolution. Skull shape evolution is much faster in snakes relative to lizards, and it's much more innovated, much more different from the squamate ancestor of both lizards and snakes. So now we haven't said anything about rates of speciation. So what I'm going to show you on this slide is the rate of speciation in snakes and lizards on the whole. And again, we see the same general pattern where snake clades tend to have relatively fast rates of speciation, and lizard clades have relatively mixed rates of speciation. And of course, I did not fairly represent Phymaturus within a chunk of things that are lialemine lizards here from Argentina and the southern cone here. But take-home message here, fast rates within snakes, relatively slow rates within lizards. There are a few lizards, yes, like South America's lialemine lizards that do have relatively fast rates overall. But snakes on the whole are much faster. When you look at the relationship between the individual clades and the speciation rate, there's really not a relationship between how fast you're speciating and how many species you have in your clade, which is, I think, surprising to a lot of people. So there are really species diverse clades of lizards that don't have fast rates of speciation. There are really relatively species rich clades of snakes that have fast rates of speciation, but within snakes there's really not a correlation. So the real difference here is that snakes have fast rates and lizards have slow rates. So I'm going to quickly go through this next set of results. Basically, what I'm going to do now is ask, for the variation that we have in traits, like let's say diet as a trait, how much of that variation in diet across all squamates comes from single nodes within the phylogenetic tree? So it's almost like doing an ANOVA. And I'm going to say, well, are there particular points in the tree that explain most of the variation in a particular trait? So let's say I have a phylogeny like this and I have some trait data. Essentially going to do an ANOVA-like analysis, and I might, for example, ask, well, if I had one way of splitting that tree, perhaps I could explain, I found a node that could explain 15% of my total variation in the trait. Or maybe I could partition it into two groups and find that, well, I have a first group that explains 15%, and there's another group that explains 15% of my total variation, and so on. I can continue on down the line. So we're going to apply this to snakes, and we're going to ask for a trait like diet, and we start splitting that tree into nodes. How much of the variation in diet can we explain by single nodes? And if we split our phylogeny into two divisions, we can explain 40% of the variation. And that's the split between snakes and everything else, right here. But if we start splitting it into more groups, we have almost into two groups, or into three groups, or four groups. These are the number of divisions overall. If we add an extra partition and another partition, it has almost no effect on the total amount of variation. So basically, to a first approximation, all of the variation in squamate diets is explained by snakes and lizards, right there. If you allow an extra group, it adds only a very marginal amount of explanatory power, and so on. That ends up being a very general result. So when we look at body elongation, almost all of it is, maybe this particular one isn't expected. That's 95% of the variation comes from snakes and lizards. But that's also true for a skull, for example, where you get about 40% from the split between snakes and lizards, and just kind of marginal increases after that. Same is true even for traits we weren't expecting, like body mass, for example. Body mass, more of the variation explained, about 35% comes from snakes and lizards. Splitting beyond that doesn't really have much impact. So we have a lot of variation in rates of things happening, but it seems like, to a first approximation, it's really explained by this fundamental split between snakes. So snakes really do seem to be different from lizards in a lot of ways. But all right, now bringing it back all the way to like one of these central questions here is, to what extent do traits like this that we've looked at predict rates of speciation overall? So we're going to look at we're going to use a multivariate sort of approach and ask for any of these individual traits, like the lability of the skull, or sensory apparatus, or their foraging mode, how much of the variation in speciation rates can be explained by any of those traits? And the answer is not very much. So this is the total amount of variance that's explained by each trait. So the most explanatory trait, the skull kinesis, only explains roughly 2% of the overall variation in speciation rate. So what we're saying here, though, is that you can still say speciation rates are really different between snakes and lizards, which they are. Split right there explains most of the variation in speciation rate across squamates. But when you try to link it to a specific trait, specific traits, it turns out that independently of that split between snakes and lizards, the individual trait effects are very small. So the best trait explains only 2% of the overall variation. So what does predict these rates of macroevolution in squamate reptiles? Well, I think what we found is that there's a huge amount of variation in speciation rate. There's a huge amount of variation in morphological and ecological rates, and there's a huge amount of variation in the amount of innovation they have. But this variation is structured phylogenetically. Clades differ systematically in these evolutionary rates. And nonetheless, though, you could say that to a first approximation, all of the variation in these rates comes down to just one thing, and that is whether you're a snake or whether you're not a snake. So, I don't know where this leaves us with the key innovation story. So, for just, I'm gonna go kind of quickly through another aspect of this story, which is looking in a little bit more detail at another facet of this. It does connect to the key innovation thing, though. So we're going to look at traits that underlie variation in rates of speciation in squamates here a little bit more explicitly. What we're going to try to do here is, so these are all microevolutionary processes that are thought to contribute to speciation. For example, we think of reproductive isolation being a key part of speciation. Ecological differentiation between populations, the rate at which populations split into species or become geographically isolated. This is sort of the microevolution of speciation. What we're going to do is ask whether we can find traits or measurements of these processes right here and ask how well do they predict these large-scale macroevolutionary rates of speciation on phylogenetic trees. So I'm going to focus in particular on how quickly new isolated populations form. And that might seem like something that's kind of hard to measure, but we think we can get to some degree we can get something of a handle on this using population genetic data. And so we're going to estimate population structure or population divergence within species and use it as a proxy or as an estimate for how fast population. And then we're going to ask whether it predicts rates of speciation. So we're going to estimate rates of speciation across the phylogeny as well. And we're going to do this on Australian sphenomorphine skinks, which is a group that I started out working on for my PhD, and we still have a number of projects focusing on them. And one of the reasons we thought this group was interesting is that they have a lot of species, we have good sampling for these things, but also they have a pretty well-defined split in terms of rates of speciation across the group. So it's essentially one group down here that has slow rates of speciation and one rate over here that has relatively fast rates of speciation. And so the prediction we're going to make is that these slow speciating lineages should have essentially slow rates of population splitting, which we're going to measure with population genetics. These lineages over here, which have fast speciation, should have relatively fast rates of population splitting. And what do I mean by rates of population splitting? We're going to do all this using RAD sequencing, but the basic idea is to look at the slope of genetic distance between individuals or between populations with respect to geographic distance. So when I say rate of population splitting or rate of population divergence, the way we're thinking about this as the way in which genetic distance accumulates with geographic distance. And so we're thinking about the slope of this relationship right here, which is essentially an isolation by distance slope, which tells us all things being equal, two populations for two different species that are separated by the same distance, could have a thing with a steep slope would be one that's accumulating genetic distance really quickly, and one that has a really shallow slope would be accumulating genetic distance slowly. So we can do this for a bunch of species, and our overall prediction is that if we're going to predict that the rate of population divergence from these isolation by distance analyses is correlated with the rate of speciation overall. And there's a good theoretical reason for thinking, and it's that species that have low amounts of gene flow are going to have high slopes for their isolation by distance relationship. Low amounts of gene flow is going to be something that we expect to be characterizations that are easily subdivided by things like landscape features, where they have dispersal biology such that they're just always splitting into isolated phylogeographic groups that can go on to form new species. So we did this for about, I don't know how many species. We did this across these phenomorphine skinks. This is the relationship we get. This is the relationship we predict. This is the relationship that we actually get. We really have no relationship between the rate of speciation and the within population divergence. Remember, we're expecting to get a positive relationship like this. And of course, each of these data points, I'm going to stress, is a population level estimate of isolation by distance from anywhere from like 5 to 50 individuals per species. So there's a lot of population level data that goes into that. So no relationship between rates of population differentiation and the rate of speciation. A similar approach to look at other groups as well, other squamate reptile groups. So one of the limitations of the study that I just showed you is that we're focusing on a relatively narrowly defined group, so sphenomorphine skinks. So maybe there's just not enough variation in speciation, for example, to really sort of tease out the signal of an effect here. So we thought, well, let's say let's go to another place and look across a whole bunch of clades of squamate reptiles that differ dramatically in their rates of speciation. So we're going to focus on 60 species of squamate reptiles from all clades, not just skinks, but everything from geckos to different groups of snakes, and ask whether we have differences in population isolation and how it correlates with rates of speciation. So these are just some representatives here, everything from coral snakes to vipers, to tree snakes, to gymnophthalma lizards, and so on. And so we use an approach that's kind of similar. Here we have this is using an older phylogenetic tree in this case. This work's a couple years old. But we did the same approach where within populations of individual species, we quantified population genetic structure in a somewhat similar way. This is just showing you the amount that the clades that we sampled, the species that we sampled, were drawn from both fast speciating groups as well as slow speciating groups from across the tree. So we have a lot of statistical variability with which we can try to tease out a signal here. And one of the interesting things that jumped out at us right away is that there are really interesting patterns of population structure or population divergence or slopes of isolation by distance. These are a couple of species of snakes here, and you can see that their isolation by distance slopes. So each of these data points here is a pairwise comparison between populations. You can see that their isolation by distance slopes are relatively slow. So these are fast rates of population divergence or high levels of gene flow across their range. And we compared that to lizards in general. We could see right away that the lizard slopes were relatively steep. So all else being equal, lizards and snakes separated by the same amount of geographic distance tend to have very different patterns of population structure in that the lizards become much more differentiated more quickly than the snakes for a given level of geographic distance. So how does this translate into an effect on overall rates of speciation? Remember, we are predicting overall that we should see a positive relationship. The more divergent you get in with respect to, the more you have genetic divergence, all else being equal, the more you expect to see a positive relationship in this regard. So these are the results separated by snakes and lizards. And here, like in the previous story, we have no relationship between population divergence and rates of speciation across this set of 59 species that we looked at. There's a clear difference between lizards and snakes in terms of rates of speciation. So snakes in blue, for whatever reason I put snakes in blue in this case, but snakes have fast rates of speciation in general. Lizards have slow rates of speciation. Lizards have more population structure, and snakes have less population structure, but they're within snakes and within lizards, there's no effect. So we have no effect overall. So what are our conclusions here? So one of the things is that in the second part of the talk, the microevolutionary processes that we've talked about, whether they're related to key traits and phenotypic evolution or key traits and processes associated with speciation and macroevolutionary dynamics, we have at best a sort of inconsistent or maybe even no effect of those predictors on overall macroevolutionary rates, even though we have really strong evidence for variation in those macroevolutionary rates. So, for example, the rates of population isolation aren't correlated with rates of speciation in snakes and lizards. And this is consistent with things that we've found in other taxa as well. So this is an example from an older study looking at rates of pre-mating and post-mating, or pre-zygotic and post-zygotic isolation evolution in Drosophila, and linking that to rates of speciation. Again, no predictive effect, despite everything that we think based on microevolutionary considerations telling us that those things should be linked. Things that evolve reproductive isolation faster should speciate more, and that's been shown in birds now too. Things that evolve pre-mating isolation associated, for example, with song discrimination should speciate faster if that is a critical engine of speciation, but we don't see a signal of it. So at the same time, when we look for key traits that predict speciation rates and phenotypic evolutionary rates, we don't find much of a signal either. So we find a relatively small amount of variation, for example, in the first part of the talk, in speciation rates that was explained by any single trait. And I think that this with what I like to think of as a predictive challenge for evolutionary biology, which is that if you were to take matched sets of taxa from any part of the world, and here I'm focusing on a couple of tree snakes, one from Southeast Asia and one from South America, and if you knew everything about these groups, and let's say, in this case, these are arboreal snakes that are camouflaged. They're about the same size. They have convergently evolved very similar diets. They're actually both specialists on eating arboreal slugs and snails. And if you knew everything about these species in terms of their ecology, their geographic history, their environmental history, their biogeographic history, you had their complete genome, you knew their life history, could you predict which one of those two species belongs to a clade that has faster rates of speciation? And I would say, turns out that despite the fact that this particular example, these species are highly matched in terms of ecology and morphology, this species belongs to a clade that has roughly 5 times faster rates of speciation. And I think the general message here is that, for me, is that we have almost no predictive ability without knowing phylogeny and without knowing, for example, that you're looking at a fast speciating cichlid fish from Africa. If you knew everything else about that species, you knew, hey, here's a fish, you got its morphology, you got its genome, you know its life history, its mating system. There's nothing about that that would tell you that it's also going to be speciating fast. The only thing we know they're speciating fast, but we know they're speciating fast by cheating because we know they're cichlids. So it's not really a fair comparison. So I don't know. I think, here's another, and thinking another is this idea of one of the key traits for, started out my talk today mentioning the pharyngeal jaw in cichlids. the key trait that people have speculated about with respect to snake diversification for a long time has been the idea of cranial kinesis. We all know that snakes have these famously flexible skulls, right? Like they can swallow massive things. So maybe that's the key innovation that underlies snake diversification. And it is true that snakes generally both are fast diversifying, highly innovative, and they have these flexible skulls that can accommodate things like this olive python here, a kangaroo. But on the other hand, what we often forget to pay attention to is that there are other examples of clades that have evolved similar kinds of traits without doing anything. This is my favorite example. This is a gecko called a snake lizard. This is actually a gecko. It's got elongate, it is legless. It is a specialized predator of lizards that lives in Australia. And it has evolved very similar patterns of skull flexibility to that we see in snakes. So this is actually a photo that I took. So I've caught this thing. I'm pushing with my thumb on the front of its skull, and it actually has this skull that is incredibly flexible, and it has this flexible skull allows it feeds on lizards that are essentially, they have bony plates in their scales, and it makes it really hard for predators to get their teeth into it. So what this thing has done is that it has a flexible skull that allows it to wrap its jaws around the skink and increase the lizard and increase the contact area between its teeth and the lizard's scales. And it has other innovations as well. So you might predict, well, here you have another evolutionary instance of flexible skulls. Shouldn't this have some big macroevolutionary consequences? But in this case, it's basically translated into almost no macroevolutionary effect. So there's only 2 species in this entire clade of things that has this particular trait. And so that's the thing we have to keep in mind when you think about these like dramatic, clades that have done these things, and they have traits like cranial flexibility. But when you look at other clades that have cranial flexibility, you can often find examples where they haven't done very much. And so, and despite this, there's still been this, changes haven't happened. There's still been this huge impact of these kinds of shifts for overall biodiversity, both in terms of the types of ecological traits that we see in the world around us, and species diversity and everything else. So the evolution and innovation have differed dramatically between snakes and lizards. And it's had a huge effect, for example, on the trophic structure of animal communities, where snakes are essentially this group that has massively expanded the amount of the diversity of trophic resources that vertebrates use, because they're these highly specialized vertebrate predators. And so, I think, maybe we like to think about this innovations. It might be that this paradigm of key innovations that we use is a little too simple. Maybe there's something more complex going on. I think that it's very possible that a lot of the big patterns of evolution actually are very dependent on context or specific historical circumstances that mean what we don't have, we're not going to have statistical predictability in the way that it's ever going to match these simple stories where a key innovation evolves and predictably causes diversification of a particular clade. Maybe that's just not the way evolution works. So in other words, shifts happen, and the impact of those shifts happening has been enormous, but it's also possible that the majority of these shifts reflect unique historical events and the unique interactions between particular traits evolving and sort of historical circumstances that mean those effects are never going to be seen again in the same way in other clades. I'm finally done. So thank you for your attention. I also want to thank, we had a mountain of population genetic work in particular, and also dietary work involving museum specimens. So I do always like to take the time to thank the museum collections that underlie so much of this work, because really without the legacy of having specimens, both in terms of tissue samples and in terms of stomach contents, we simply wouldn't be able to do this type of work. So happy to hang out and take questions. So thank you.
Bueno, ¿alguna pregunta? Sí, Diego.
Thanks for the talk. Really thought-provoking. So, what can we say about what determines speciation and speciation rates? Does this tell us something about the role of neutrality
The role of,
or what? So
oh, the role of, tells us about the role of
neutrality
neutrality.
in evolution. So traits don't really matter, or does it tell us something about our poor understanding of the functional role of traits? So maybe we think we
Right.
understand what traits mean, but they don't really do what we think they do.
Right. That's a good question. So I will answer this with my bias explicitly, which is that while I think neutral molecular evolution is like a thing, obviously there's lots of changes that are happening that are effectively neutral. I don't think any of these phenotypic type changes. I don't think there's any neutrality. I'm a hard selectionist and a hard adaptationist from that sense. But so it doesn't. I don't think there's any implication that any of this is neutral changes in any way. But what I think it is more getting at is that, and I agree very strongly with the second, which is that we don't know. Not only do we not know in some cases how adaptive particular things are, but we don't know in what way they're adaptive, and the fact that they are also differently adaptive for different clades and in different contexts. And I think we definitely have a very poor understanding of that. So I would agree with all of that. But this isn't really, even beyond that sort of spiel, this isn't really saying anything directly about the extent to which, it's not implying anything about adaptive and non-adaptive. It's just saying that the simplified view of trying to pick out a single type of trait and look at its effects independently across different clades. Well, those effects are generally, pretty marginal at best. So it's that could be true whether they're adaptive or neutral in this sense, but I don't think any of the traits that we're looking at, I mean, I think all of even these sorts of traits, I would say we have enough of an understudies, not that I'm not mentioning here, but any of those morphological traits, any of those dietary traits, those foraging traits, there's a lot of evidence for the adaptive significance of those traits within different sorts of contexts. I think they're all functionally relevant traits in general. Even things that are, like vertebral counts for, I mean, there might be places where it doesn't matter so much, but we also know that there are, like in snakes, for example, sometimes even subtle differences in numbers of vertebrae can affect, their locomotor speed, for example. So, which has obviously ecological consequences for predator escape and so on.
¿Alguna otra pregunta? Sí, no sé si, bueno, igual, Pau, dale.
Thank you for the talk. So huge, it's a lot of information. And from all of that, I was wondering if you found a relationship between variation of the skull and variation of the diet.
Between variation of the skull and between the skull, the shape of the skull, and the diet.
Yeah, morphological variation in general, and dietary variability, and if you consider one drives the other.
I mean, we didn't look at that in this case, but we have looked at it in other studies, and a lot of other people have looked at it. I mean, I would say in general, it seems clear that within reptiles, for example, their morphology is tracking, whether it's their body shape tracking habitat or foraging mode, or whether it's skull tracking what they're eating. I mean, I think there's a really strong overall signal of those changes happening in response to shifts in ecology, diet, for example. So, I mean, we didn't look specifically at it here, but so I guess I would say I guess I would say that I feel that the evidence is consistent with a lot of the changes a lot of the variation you see in skulls of squamates being driven by changes to different kinds of ecology. That's going to depend a little on which clade you are, but I think overall there is if you look at the degree to which snake skulls, for example, are specialized in different ways for catching prey or for subduing prey, I think the evidence, there's a lot of evidence that they have shifted in different directions depending on what they're eating. In some cases, you also have habitat that affects the way those skulls look. So the snakes and lizards that burrow and go into like cracks in the ground, they have different kinds of skull geometry that are all making them more robust. So there are other things going on too, but I do think that ecology, that ecological effect is huge. Does that answer your question?
Are there cases from other groups suggest particular traits can explain the diversification, or this unpredictability is a common pattern in the tree of life?
So I will give my complicated answer, medium complicated answer, which is that you can look in the literature and find the claim that there are specific traits that do in fact predict diversification in general. And there may be a couple of them that might hold up, but I would say this literature is also extremely contaminated with statistical problems, like to a level we will not go into right here. But I will say that I believe most of this work, and I do not believe most of this work. So I would say that overall, the vast majority of things that have strong a priori reasons to think they affect stuff. Here's a great example. Sexual selection in birds is one of the things associated with sexual selection in birds. There's a strong reason based on theory to think that should drive faster speciation. Sexual selection in general should lead to faster evolution of mating signals, more divergence per unit time in recognition characters that causes more pre-mating isolation, all else being equal, and you should get faster rates of speciation. But that has been, there is yet no strong evidence comparatively with speciation rates linking those. So there were, some studies have found it, then studies came along and said the methods those studies used were problematic, and then other people used different data and different methods and showed that the effect wasn't very strong. So almost in most cases, I would say that there is no real overarching effect. It doesn't mean it's not involved in some particular clades or others, but what it does suggest is that the simple ways in which we're approaching things is saying, there is in the case of birds, this is usually what it is. There is a simple trait called sexual, and we can quantify it by how much dichromatism there is in the plumages of between male and female adult birds. Well, there are like multiple problems there. There's the assumption that that's a good index of sexual selection. There's an assumption, for example, that species of birds that have no sexual dimorphism are having less sexual selection than the ones that are fully dimorphic. And some of those assumptions can break down, and I think we haven't done a good job of thinking about that sort of like clade-specific complexity. So I would argue the sexual selection case there's no good evidence for it comparatively. It also hasn't been as well tested as it could be for that reason. So I guess coming back to your question, I personally think there are key traits that do have some predictability, but we are often thinking about those traits in too narrow or too clade-specific of a way. So if you think about like craniokinesis, for example, as like a trait, well, what if that trait, that is one have figured out how to become more ecologically labile or versatile evolutionarily. All else being equal, a snake can evolve more in terms of its ability to use different prey types, because snakes have figured out developmentally or genetically how to have more evolutionary faster rates of skull evolution, and so that they can just access resources that other clades can't. So maybe by that logic, focusing on the skull shape is the wrong trait. Maybe what you should focus on is the trait is ecological lability in a general sense. And by that metric, you would compare snakes, and then you have craniokinesis doing and other things that snakes have done, and then you also have pharyngeal jaw and cichlids, which serves the same role, but is a completely different type of trait. So maybe we have to think differently about traits and think more broadly before we can really sort of test these things, and rather than focusing on specific kinds of traits associated with particular clades. Like, I think it's still an open question whether sexual selection predicts speciation, but I think that we generally focus on very narrow clade-specific definitions of it. So that kind of answers your question.
Yeah, because when you showed the bars, the percentage of explanation, I thought that maybe if you look for a synergic interaction between the traits, maybe the bar will be.
I do think it's possible that at this, here's another way of saying what I would say that I think it's entirely possible that there is if we look across squamate reptiles, so there's 10,000 species of squamate reptiles, there's 10,000 species of birds, there's 30,000 species of ray fin fishes. I would say that at the scale of even within any of those clades, that might not be a big enough scale to detect the effects here. Because if you look at how many actual, big shifts in rates, for example, in macroevolution have happened within squamates, it's really only a couple that explain most of the variation. It's really, to a first approximation, there's really only one big data point there, and that's the snake pattern versus the lizard pattern. I mean, yeah, there are some other shifts going on within lizards and some other shifts going on within snakes. But clearly, if that much of your effect is at that sort of scale, that might mean you need to go all vertebrates or even like all insects. And so you might need to go to much broader scales. But then also, that question, you need to think about types of traits that have a meaning in terms of their function and their functional significance for those clades that can be compared across completely different kinds of organisms. So, what is that? What is sexual selection within beetles in terms of an indicator trait that gives you a comparable metric of sexual selection intensity that also lets you put them in the same universe of statistical comparisons as birds and ray fin fishes? If you answer that question, and you can do that, I think we can possibly find ways of getting at predictability in that sense. Because I do think once you get to that scale, there's going to be something that predicts this stuff. So even if it's context dependent, there's still biology that's going on. The clades are doing something different. Whether we can actually get there at the right scale, I don't know. You have a question too, right?
Just a short one, I think. Do you deal in some way with random events regarding, I don't know, ice ages that can perhaps affect the evolutionary speciation events? How can you deal with them to the future, in predictions to the future?
How can you
include them in the analysis?
Like, how do we incorporate them in the analysis?
Yes.
I mean, essentially, we don't even level of detail just because there's such a. I don't think it's practically feasible on one level. But there are multiple about that question. One other way, though, of thinking about it is that to some degree, some of that isn't going to matter very much because, like, let's say Pleistocene stuff, those speciation rates that we're estimating here, with the methods that we're estimating them, are really sort of almost like an averaged effect across the tree that is. I think about them as like basically it's almost like an average at any given species. It's sort of like an averaged rate that takes into account sort of the last like 10 million years of history of that lineage. So it's averaging over a lot of noise that's happening. Like you might say, well, what did the last glacial cycle do? Well, that rate is going to be averaged over a much. Now, if there had been a clade where a whole bunch of species had formed in the last 20,000 years because of glacial fragmentation, that definitely would have affected things. But we don't really, certainly within squamates, we're not really seeing that. But if you were like, hey, I think, stickleback fishes, which are supposed to go really fast speciation, and Dolph Schluter's work in post-glacial lakes in Canada, if you took every one of those like stickleback populations because they have some reproductive isolation in a post-glacial lake, and you said, I'm going to call them species and put them in my tree, even though they've all arisen within the last 4,000 years after the glacial cycle, that would dramatically change results. It would also involve lots of other weird things that how we conceptualize species, but there are ways in which it could matter and where it could matter for particular groups. But I would argue that these results are sort of at a scale where some of that stuff kind of gets, average. Yeah, it's definitely a source of noise and bias potentially.
Thank you.
¿Alguien más? ¿En el Zoom? ¿Hay alguien? Perdón. Pensé que. A ver. No. No. ¿Alguien más? Bueno, muchas gracias.
Gracias.